never quite sure when you walk in the room if it's really going to be anybody here. So thank you. So what I'd like to do today is make it sort of neat and tidy, is to try and get you down as far as the stomach. We won't do the stomach today, but I'd like to get the food, the description, the information down as far as that. So what that is going to involve is covering, I hope, these three topics here: the taking in of food and water, how it's regulated. I said I wasn't going to say much about that, but it's something that needs to be talked about briefly. The role of the main glands associated with the mouth region, the saliva glands, and then how we get the food once it's being chewed and mixed with saliva, how we get it down into the stomach, swallowing. So let's start off with ingestion, taking in of food and water, and not how we do it, but more why we do it. So the questions to ask here are really very simple ones, but the answer is not so simple. The questions are: what makes us feel hungry? Why do we say we have a good appetite? What makes us feel thirsty? And how does the body regulate these things? The first thing maybe to say is that hunger and appetite are not exactly the same. We tend to use them interchangeably, but strictly what hunger means if you're hungry is that you need the food. Whereas appetite is more you want the food. So, you have your meal of meat, two veg, salad, etc. because you're hungry. And then you have a nice big piece of chocolate cake afterwards. Not because you need it, but because you want it. So, with that in mind, let's look at this balance equation here. Energy in equals energy out. We take food in, which is obviously providing the energy, and that is converted into these things here. We use the energy for work, it produces heat, and anything that's sort of left over and not used at that moment is put into some storage form. So what influences this balance? There's work being done on this for a long time, and some of the early experiments, you don't have this, I don't think you copied it. People did a lot of the early work, of course, in animals, in rats in particular, and what they did in this particular case was to show that a normal rat, normal adult male rat, is about 400 grams, and this poor animal or its little mate was exposed to various stimulations or lesions. In lesions in the hypothalamus, you can alter its weight very dramatically. So this poor creature is weighing over 1 kilogram because certain regions which we call the feeding center in the hypothalamus were stimulated, or the same sort of result was obtained if you destroyed, made lesions in another nearby region, also in the hypothalamus, called the satiety or fullness center. So something up there is doing the control. But we need to then consider what the inputs, physiological inputs to these centers. Obviously this is, it tells us something, but in an experimental sort of unreal situation. So in real life, what is actually going on? So these centers, this is picked up in figure number 13, what we now know is that the hypothalamus contains the so-called feeding center, which says, go ahead, you need to take in more food, and a satiety center nearby. Here's the regions, the lateral and ventromedial regions of the hypothalamus. The satiety center says you feel full. But what inputs to them? So here at the bottom of this figure are certain factors, and I know, again, I'm going to be saying this is an extremely complicated area. It's very important, very, very interesting. We certainly don't know the whole story. And somebody a few people came in, there are some handouts of the next figure, maybe you can get it afterwards. But I've added, because what's here, you just read a list, but I don't think on second thoughts when I was thinking about this, I don't think it gives you the picture of how they're working together. So let's just look at this first. Factors which affect the intake of food are usually divided into orexigenic factors, and that's maybe a word that you think you've never heard or haven't come across. What does it mean? But you do know probably anorexia, anorexigenic or anorexia. A person with anorexia is somebody that almost stops eating, they dramatically reduce their food intake, and so anything that reduces food intake is called an anorexogenic factor, and the opposite of that, the ones that increase it are orexigenic. And here's a list of them. A hormone grading that comes from the stomach, something called neuropeptide Y, MPY from the hypothalamus, maybe other ones too, lots of question marks around here. The ones that decrease food intake seems to be some on a longer list. Something from adipose tissue, leptin, a hormone, insulin, which you do know about in pancreas, other ones from the intestines, and so on. So I'm going to come back to those, and that's what that extra figure is. But let's just sort of almost get rid of these ones at the bottom. Usually if you ask somebody, why do you feel hungry? Oh, my stomach's growling, it's empty. Why do you feel full? Oh, my stomach's distended. I just don't want to more. And that's probably part of it. So these miscellaneous factors down here, things like distension, temperature, you tend to eat more in the winter time when the ambient temperature is lower than in the summertime. And it's too hot to eat and can't be bothered. Glucose utilization by cells, that seems to make sense. That cells are utilizing glucose well, and they've got good supply. It's essentially giving you the message, okay, you've just had a meal, cells are happy, you don't need to eat anymore. And these contribute. Even things like social environment. Somebody sitting next to you brings out something, you think, that smells really good. And until that moment, maybe you hadn't thought you were hungry. Or you look at the clock, it's a bit early now maybe, but if it was just coming up to noon, or I don't know, 6 o'clock at night, and you've been busy doing something else, you've forgotten about food, you look at the clock and you think, oh yeah, I am hungry, it's time to eat. So these are important, but it's not really the main physiological aspect of this topic. So what I've done in this figure, which you can call figure 13A, is to just put into a sort of semi-realistic form these factors here under 1 and 2. So what you're looking at here at the top is where these things come from. And these are hormones or hormone-like molecules. Peptide YY, gradient from the stomach. This one from the intestines, small intestine linear, insulin from pancreas, and the hormone leptin from adipose tissue. And of these, I've used green for the ones that say, stimulate, go ahead. For you, they're just the solid lines. The red here is inhibitory. The dotted lines are inhibitory arrows. So when you don't eat a meal for a long time, ghrelin is released from the stomach, and that works by traveling to, all of this is the hypothalamus, these blocks here. This is actually the arcuate nucleus, if you're familiar with that, in the hypothalamus, where there are regions where this another peptide, neuropeptide Y, is released. So for example, ghrelin will increase the release of neuropeptide Y. Neuropeptide Y in turn acts on a nearby region, the eating center that we talked about in rats, and that will cause you to make you feel hungry and will increase your food intake if possible. So this is a relatively short-term control system. Because as the meal comes along and goes into the intestines, you can get other peptides released, other hormones or hormone-like molecules released, this peptide YY. The food starts to pass down the system, and this has an inhibitory effect on what you can call the accelerator. So what that's saying to your body is, I, the body, know that you, the body, have just had a meal, because there's food coming down into the intestines, and it's causing release of this peptide YY. So you've had a meal, you don't need another one just yet. So it inhibits what you can think of as the accelerator. Again, as the meal goes down into the intestines, what we'll learn more from Dr. Harvey is that insulin is released from the pancreas. And this can also inhibit. It has a double effect. It inhibits the accelerator at the same time as stimulating what you can think of as the brake. This melanocortin is another hormone-like molecule which comes from other regions up in the hypothalamus. And this brake can act on the satiety center. So you've got things which stimulate the feeding center, things which stimulate the satiety center, which decreases the food intake, and there's probably a lot more arrows than I put in here. Things that can stimulate the feeding center can at the same time inhibit the other one. Now, on a longer term basis, probably one of the most important of these is leptin. This is, in a sense, a measure of how much fat tissue you have in your body, how much adipose tissue. The more that you have, the more deposits you already have, fat in a storage form. What the body is signaling, or needs to signal, is to say, look, you've got all this fat, maybe you should cut back on your eating. And so, these ones are going to be acting more on a meal-by-meal basis, presence of food in the fat. This is more a long-term now. Leptin will inhibit the accelerator and probably also stimulate the brain. So, I don't know if this is something that's going to help you sort of put together. These are just the basic facts here, and this is trying to put them together a little bit. And so if anybody didn't get one of these, you don't need to draw it out. I've got a few more here. Okay, a similar idea is how we regulate water intake, and I think this is probably more familiar to you from things that you've learned both in your hero lectures and also in your cardiovascular lectures. So regulation of water intake, being two main factors that stimulate this or that affect it, are changes in the osmotic pressure of the extracellular fluid and changes in its volume. Is our extracellular fluid more or less concentrated than it should be? Do we have more or less of it? The one that we're most sensitive to is changes in osmotic pressure. It only needs a very small change. The osmoreceptors detect this change are present also in the hypothalamus, and this will send a message to a thirst center, again hypothalamus, and you will go out and seek increased fluid intake. Less sensitive, but also very important, changes in volume affect various stretch receptors that you will learn about in the cardiovascular system. Again, inputs to the thirst center, and also from your kidney lectures. The kidney will alter its release of the enzyme renin. Renin acts on angiotensinogen and releases angiotensin II. And angiotensin II is a very potent stimulator of thirst. So these working together can help to keep osmolarity and blood volume, extracellular fluid volume, within the normal range. Okay, so that's all I'm going to say about that topic. There's a lot more to say, but we just don't have time. And I want to move on then to the next one, which is saliva glands, production of saliva. How do they do it? Why do they do it? And what's the significance? So, before we wrap up, we have three main pairs of salivary glands. Just a bit of nomenclature. The largest glands are the carotid glands, and these are the ones that are sort of the angle of the jaw, sort of near the ear. And if you've ever had mumps, these are the ones that tend to get very swollen and to be extremely painful, and you're very aware of where they are, somewhere back up here. The second largest ones, and these are all paired on the left and right hand side, are the submandibular, sometimes known as the subaxillary glands, and then the third ones are the sublingual. And these two, the submandibular and the sublingual glands, are found underneath the tongue, in this little region in the throat here. And these glands, as I say, they're paired. They have ducts, so the glands make the saliva. You see how they do that. This saliva flows into a system of ducts. The parotid duct opens into the mouth somewhere up near the top back teeth. The submandibular and sublingual glands, they have ducts which open near the front of the mouth under the tongue. And if the next time you're looking in a mirror, you lift up the front of your tongue on each side, you can see some sort of, looks like pimples. These are the openings of the submandibular and sublingual ducts. Okay, focusing in on the histology, the more detailed structure, figure number 15. The plain picture that I think you need to get for salivary glands is that they're a bit like a bunch of grapes. And this is a type of structure that you see again in the pancreas, and also when you have your reproduction lectures, the mammary glands. Also, general bunch of grapes type of arrangement. What do we mean by that? Well, there's different types of salivary glands, and they've got different structures. They're not all exactly the same. But if you take a generalized salivary gland and look at individual cells, you see something like this. The cells which produce the saliva, the ones I've done in yellow, are called acinar cells. And these acinar cells, that's referring to a single cell, are grouped together in clusters, each one called an acinus, and this would be the grape. And there are a couple of different types of acinar cells. These ones here are called serous acinar cells, and they produce a fairly thin, watery secretion, which has got water electrolytes, maybe some enzymes too. We'll talk about that in a moment. Whereas the mucous acinar cells, as you might guess, produce a thicker secretion with mucus present in. And sometimes these are in separate acini, plural acinus, and sometimes you can have mixed acini. I've shown them separately here, so serous and mucus. Once the grapes, the acini, have produced their secretion, it flows into a system of small ducts, which then go into larger ducts, and larger still, until ultimately they go into the mouth. So these duct systems are the stalks in a bunch of grapes. The acini, the grapes, the ducts are the stalks. Another type of cell, the ones that I've done here in blue, around the outside of some of the acini, are smooth muscle-like cells, myoepithelial cells. And they're able to contract and relax, as you'd expect for smooth muscle-like cells. And they don't themselves produce saliva, not like the acinar cells, but they probably have a role in squeezing out the saliva, which is produced here. When these contract, they're going to squeeze down on the grape and push out whatever secretion is present. The other thing to notice about this is the blood supply. It's a bit unusual. Blood is coming in here and breaks up into, so the arterial inflow here breaks up into a network of capillaries around the ducts. And then you have portal vessels here, which then lead to a second capillary bed network around the acini. So you know, portal-like vessels are ones which connect two sets of capillaries, and you see these here. So What is this telling us? What's the significance? Well, one thing I think it suggests, when you've got a rich capillary bed, it's suggesting that the regions which are supplied by this, the ducts, number 1, and the acina cells, number 2, are both important in the production of saliva. And that's what we'll see in a moment. That both the acina cells and the ducts are very important, for they work together in the production of saliva. I'll come back to that in a moment. First question then, what is saliva and what is its function? There's nothing esoteric about this. They're all pretty straightforward, and you could probably ask somebody who really knows nothing about physiology, they haven't done it, and they can make a pretty good guess as to some of the things that saliva does. What is it? It's mostly water. It's got electrolytes in it, relatively high concentration of potassium bicarbonate, and relatively low concentrations of sodium chloride. I pause there just in case somebody wants to put their hand up and say something about that. Any thoughts? What type of fluid is saliva? Extracellular fluid. Right, someone heard that. Does that give you a clue? Opposite way around. Normally you would expect extracellular fluids to be the other way around, but this is what you see. So normally you'd have relatively high sodium chloride in extracellular fluid and relatively low potassium bicarbonate. Why? I'm not sure of all the answers to this. I can tell you, and I will tell you in a moment, how this occurs, but why? Well, you can think of a couple of reasonable answers to that. And one is that the relatively high bicarbonate is a protective aspect of this. We don't want acids eating away the enamel on our teeth. There could be acids in food, acids coming back up from the stomach if something goes wrong. And so a high bicarbonate, by neutralizing the acid in the mouth, is protective. That's perfectly reasonable. Another reasonable suggestion is that the relatively low concentrations of sodium chloride enable you to taste salty foods more easily. If you had a really high concentration salty stuff in your mouth all the time, then when you eat foods containing sodium chloride, there have to be really high levels for you to detect that on top of the large amount that was in your mouth. So by having a low background sodium chloride, it means that our ability to detect salt in food and therefore adjust our intake accordingly is enhanced. So I think those are two of the answers there. Okay, saliva's also got glycoproteins, mucins. In mucus is mucins, which are the glycoproteins, plus electrolytes, plus water. There are some digestive enzymes. I've only mentioned one here because it's the best known one, and that's salivary amylase. It's not crucial to life. It's not crucial, really, to normal digestion. It's there, it normally works, it does something fairly important, but even if we don't have the salivary amylase, we can more or less say, so what? Because we have a pancreatic amylase lower down, which can take over the function of this one. We have a whole bunch of other components in saliva. Although it's mostly water, it's not a boring solution. There's lots of things there. A lot of them we don't know what they're doing there. Some of them we do. Lysozyme, immunoglobulin A, again, having some protective functions. Growth factors, something called nerve growth factor, and apparently the highest source in the body of this nerve growth factor is the submandibular glands of male rats. But that's where it's discovered. Why there's so much, what it's doing. Kallikrein, I would emphasize that. You know, at least a couple of you are thinking about kallikrein these days, or going to be. So I won't say any more about that now. And other things. Okay, functions. These are the ones that you could probably guess. If something goes wrong with your salivary glands, and they're not producing adequate amounts of saliva, as I just said, you probably won't experience problems with digestion. But what you will have problems with are things like oral hygiene, because of the simple things that the saliva is doing, as stated here. Things like tasting, you have to dissolve things to taste them. You try swallowing when your mouth is very dry. It's difficult. This is why I usually know public speakers, they have a carafe of water on the podium, because maybe they're nervous, and when you're nervous, your saliva production is reduced, and it's very difficult to talk, swallow, etc. And then I will talk in a moment about the role of this enzyme in digestion. Okay, let's look first of all at how saliva is produced, mechanisms of production, and this I hope will explain why it has the electrolyte composition that it does. So I'm looking again at a slightly simplified diagram here, figure number 17, showing an acinus and a single duct here, duct capillaries and acinar capillaries up here. Production of saliva is a two-step process. This is something, in a way, you're already familiar with. So from the kidney, you have filtration, you have certain composition of filtrate immediately after it's been filtered, but as it travels through the nephron, in various regions, some things are added, some things are taken away, and you end up with something that's quite a different composition. But the details here are very different, but that basic idea also applies. The acinar cells produce what is called a primary secretion. And then as this primary secretion moves down to the ducts, it becomes what its composition is modified. So what's actually happening? The acinar cells secrete water, which contains these ions here, nitrogen, sodium chloride, potassium bicarbonate, essentially in the same concentrations as they are in plasma. So the initial, the primary secretion is isotonic. Now I haven't gone into details as to the exact ionic mechanisms here. I think maybe this gets a bit too much, but if anybody is interested, I did bring some figures if you want to know, well, what ion channels or what transport systems are involved. I've got that there. Let's just say they go through appropriate transporters and pumps and all the rest of it, and you have an isotonic solution here. As this moves down the ducts, as shown here, what happens is that sodium and chloride are reabsorbed, not all of it, but some of it, from the duct lumen back into the blood. At the same time, potassium and bicarbonate, more of the potassium and bicarbonate, so in the opposite direction, they're secreted from the duct capillaries into the duct lumen. And the size of the arrow is roughly indicating the relative amounts of transport taking place. So what this is saying is that more sodium and chloride are reabsorbed than potassium and bicarbonate are secreted. So you're taking away, if I just put a number to it, this is a completely random number. Say you took out 100 units of sodium chloride and added back 80 units of potassium and bicarbonate. The solution, the mixture in the duct would then be more dilute than it was up here. And in fact, this is what's happening. The saliva that is produced is always hypotonic, more dilute than planned. You might be wondering, wait a minute, sodium chloride being reabsorbed, making the kidney, if sodium is moving somewhere, water tends to follow osmotically. What would stop the water moving here? It doesn't move here. Impermeable, yes. These ducts are essentially impermeable to water. So water stays behind, take out more ions, you add a small number of ions, so you end up with a hypotonic saliva, which is relatively rich in potassium and bicarbonate, because you've added more here, and relatively poor in sodium and chloride, because you've taken more of those away. And again, if anybody wants to know iron channels and so on in the ducts, I've got that here. Okay, how is this regulated? Usual questions, or the sort of pattern that I'm going to be following, is to say, what a gland looks like, what does it produce, maybe how does it produce it, how is it regulated, and why is it so important. So regulation of salivary glands, as far as we know, is under nervous control only, at least definitely in the short term. And I'm not going to talk about other possibilities. So let's say, when you eat a meal, salivation occurs, and that's due to changes in nervous activity. And the top part of this figure is showing, and the bottom part summarizes what I'm going to be saying, is that you can relatively easily measure the production of saliva. You can put a little tube through the opening of the duct in the mouth. You can measure drops of saliva as they come out. At least in animals, you can also relatively easily measure blood flow. Why is this important? Why do you want to measure blood flow? If you're producing a fluid from a gland, where's that fluid coming from? Ultimately from the blood. So if you put out a large volume of fluid, you need to have an input of a large volume of fluid. You need to increase your blood flow. And salivary glands in humans, I forgot to mention this, but they put out about, on one of the earlier figures, maybe a litre or so every day. And somebody once did a measurement in cows. Cows when they're in the field or whether the barn, they're sort of slobbering. I think they measured it something like 50 gallons of saliva per day per cow. Anyway, we're not talking that large volume. Okay. So what changes salivation and what changes blood flow? If you stimulate electrically the parasympathetic nerve to the glands, you get an increase in blood flow. When it goes up, it's an increase. So it's a quick and large increase in blood flow, and you get copious salivation. If you activate the sympathetic nerve, remember from your autonomic lectures, salivary glands have got double innervation. If you activate the sympathetic nerve, the blood flow mainly decreases, and there's a small amount of saliva. Does anything about that strike you as a bit unusual? No? Sympathetic innervation usually speeds things up as a suggestion. You're sort of halfway there, yes. The sympathetic actually does what you would expect it to do in this case. Again, I'm not sure, I think I'm trying to emphasize this in autonomic, and probably Dr. Kopinski, when he talked in cardiovascular, told you that in most or many blood vessels in the body, sympathetic causes vasoconstriction, which is what it's doing here. Constricts, you get reduced blood flow. Yes?
Well, not never mind.
Hmm?
I was going to say, do comparison to
Normally, you're right, parasympathetic normally doesn't, well, I should say normally, often doesn't do anything to blood flow. But I remember on a chart or a table I gave you, I said it's a very complicated area, and that sometimes, I said parasympathetic sometimes does. This is an unusual situation. It's a very dramatic effect. It's not one of these things you think, oh, maybe it's doing something, maybe it's not. It's very dramatic, and parasympathetic increases blood flow. And it needs to, because parasympathetic also dramatically increases salivation. Salivation with the sympathetic is minimal. In part because, at the same time as it's trying to produce saliva, the blood flow is reducing. And you can't put a lot of fluid out if you don't have a lot of fluid coming in. So this is sort of the dry mouth situation. Now, if I told you that if you give, okay, parasympathetic releases certain transmitters, and the usual one, acetylcholine. Acetylcholine acts on what type of receptors would we be talking about here? Muscarinic, thank you. So if you give a muscarinic antagonist, a drug called atropine, after atropine, that's what you might expect. Not actually what you get. Salivation is blocked, but the increase in blood flow is not. Any thoughts on that? Yeah? Sorry, I'm really kind of hear. I'm not sure, I might have missed something. Another try there, yeah? Okay, carry on from that.
It inhibits the gland directly.
It inhibits the gland directly. Okay, so you're bringing in nicotinic receptors, not quite, but you're getting sort of an idea of different receptors, yeah? Yes, thank you. It's using probably different transmitters. Remember, non-adrenergic, non-cholinergic. It's an unusual situation, but it's, I wanted to point this out because it's here. So salivation is blocked by a muscarinic antagonist, and it's probably a very straightforward ACH acting on muscarinic receptors. Increase in blood flow is unusual to have in the first place, but it's doubly unusual because it's not blocked, as you might expect, by a muscarinic antagonist, which suggests, as you quite rightly said, that there's other transmitters involved, and we won't go any further than that. At least if you want to talk about it, we can do that after. Okay, very briefly, the main enzyme in saliva is amylase, and it initiates digestion of carbohydrates. In our diets, one of the main carbohydrates that most people have is plant starch, which is a mixture of amylose and aminopectin. And please forgive me if you've all taken biochemistry and you know far more about all these things than I do, but just in case some people haven't. Amylose is made up of a straight chain of each of these black dots is the glucose unit. These are joined by 1,4-alpha bonds, straight chain. Aminopectin, similar, but in addition, it has these branching points, which are 1,6 bonds. So you've got a mixture of these. Animal starch or glycogen is sort of similar to aminopectin. Salivary amylase comes along, and it gives a partial breakdown, partial digestion. And what you get is you get disaccharide, maltose, you get trisaccharide, motor triads, and it does not attack the branching points. Amylase cannot attack these branching points, so they remain, and you get what are called alpha-linodextrins or oligos, meaning a few oligosaccharide, probably some about 8 or 10 amino acids there. So it's not a complete digestion, which would be to glucose if it was completely digested. It's a partial one, and we need more to happen for that comes later on. If you don't have salivary amylase, pancreatic amylase can do essentially the same thing. Okay, now I need to talk about swallowing. How do we actually get the food down into the stomach? Well, first of all, just to a bit of terminology here. The pharynx is the region right at the back of the mouth. This leads into the esophagus, which in turn leads into the stomach. Now, I said last time that most of the muscle that I'm going to be talking about is smooth muscle. But there were two main exceptions at the very beginning and at the very end of the class. We look at the beginning here. So this muscle up here is skeletal striatic muscle. Going down up the top third or so of the esophagus is that type of muscle. The lower region and the stomach and all the way down right to the far end is smooth muscle. And there's a transition zone in the middle. Now, if you take some pressure measuring device and insert it into here and measure pressures here, what you find is that you've got a sub-atmospheric pressure in the esophagus. And what that means is that there's a tendency, because things move down pressure gradients, that you could have air, a lot of air going down this pressure gradient into the esophagus and then ultimately into the stomach. Okay, it's not the end of the world, it's not that dangerous, but it's not something that's desirable to happen. More seriously, you could have stomach contents going up. From this is higher pressure, they could go up here, and that's potentially extremely dangerous because stomach contents are highly acidic, and you don't want them in the stomach. So to help prevent this, we have sphincters, an upper and a lower esophageal sphincter, which are high pressure regions, and these remain tonically contracted, closed, most of the time. So when we talk about swallowing, how we do that, it has to, in part, explain how we can, on a temporary basis, open up those sphincters. And this rather complicated figure shows you number 1, 2, 3, 4 different phases of swallowing. Maybe just start by telling you what those phases are. We'll just come back to that. To make it a bit easier to describe, swallowing is usually divided into 3 phases. The oral phase, when food is in the mouth. The pharyngeal phase, when food is in the pharynx. And the esophageal phase, as it moves down into the esophagus. And in reality, they merge into each other, but it makes it easier to describe what's going on, to do it like this 1, 2, 3. So the oral phase is when food is in the mouth, and that's this number 1 here. So the green, the stuff that I call in green, is the food which is taken in, you've chewed it, you've mixed it with saliva, and it's ready to be swallowed. And what happens when you swallow, you use your tongue to push the food rollers to the back of the mouth. This is where the pharynx is. So we're gradually moving over here. The food is moving here into the pharynx. So we're moving from the oral phase, which is under voluntary control. You can say to yourself, I've chewed this thing 100 times, I'm tired of it, it's time to swallow it, push it to the back of the mouth. But once it reaches this region here, the pharynx, and stimulates certain receptors there, touch receptors, from then onwards, it becomes involuntary for most people anyway. And what happens is that when it reaches the pharynx, this causes the soft palate at the back of the mouth to close off the nasal passages, so the food doesn't go the wrong way into the nose. The front of the tongue should probably be raised up a bit more hair, front of the mouth to stop the food coming back out of the mouth. And then what happens is you get waves of peristalsis. This is muscle here. And touch of the food against these touch stretch receptors sends a message up to a swallowing center up in the medulla. And this will send a message back down again, and you get a wave of peristalsis moving down here, which pushes the bolus ahead of it, further down. As it goes down, this little flap here, the epiglottis, closes over the entrance into the trachea. So the food can't go into the nose, it can't go back into the mouth, at least not easily. It can't go, we hope, into the trachea. And the path of least resistance is to go through the upper esophageal sphincter, which is a temporary relaxation, allows the food to go through, and it goes into the esophagus. So it's coming from the pharynx through this, which opens into here, and this wave of peristalsis will continue to push it onwards here. And then ahead of this, you need to have a relaxation of the lower sphincter. I'd like to just squeeze in just a little bit of information here about the lower sphincter. It's kept tonically contracted most of the time. Obviously has to relax to allow food to go through, but you want it to be tonically contracted so that the stomach contents don't come back up again. So there's tone in that lower sphincter. The more tone there is, the more it's contracted, and the less gastric contents can come back up. If the tone decreases, it's relaxing, and reflux can occur. And this condition of gastroesophageal reflux disease sort of highlights this. So what are some of the factors that may lower the tone of the lower esophageal sphincter? Then you see there. Think of it this way, maybe next week you're on holiday, you go out for a good meal somewhere, maybe it's an Indian restaurant, you have a nice hot spicy meal, maybe you go somewhere who's serving fish and chips, fried foods, onion rings, garlic, or you go to an Italian place, nice garlic and tomato sauces with red peppers. You follow this up with a chocolate cake, some after-dinner mix. You think you're going to have something healthy, so you have some fruit, something to drink, tea, coffee, a couple of beers, maybe you're on a pill, by this time you've got a headache, you shouldn't, but you go outside and you have a cigarette, your exams are coming up again, you think you better run it off, or you just go to sleep, whatever, and then you have this horrible pain in your chest. And you get, not the last matter, you've never had it, you wonder is it a heart attack? It's called heartburn. And it's probably due to this lower tone of this sphincter. And as it says here, consequences, the highly acidic gastric juice from this mixture comes back up again. The stomach, as we'll see later on, has mechanisms for protecting itself against that acid because it sees it all the time. But the esophagus doesn't, and it can cause damage to the lining, inflammation, stimulation of nerve endings, and pain. So, think about that next week, and I'll see you.